Light reactions of photosynthesis. Light-dependent reactions take place on the thylakoid membranes. And what's the purpose of the light-dependent reaction? Well, it's to produce ATP and NADPH for the Calvin cycle. It's also where water is split to make oxygen. Now, light reactions, they involve pigments like chlorophyll contained in large proteins called photosystems. These photosystems absorb photons or sunlight. There's two main photosystems that you need to know, photosystem 2 and photosystem 1. So photosystem 1 is known as P700 because it can absorb light that is 700 nanometers in wavelength. Photosystem 2 is known as P680 because it can absorb light at 680 nanometers. Now, a way to remember this is to think of these numbers without the zeros. So P700 would just be 7 and P680 would just be 6 and 8. Photosystem 1 has one number, so it's just the 7. Photosystem 2 has two numbers, so it has to be 6 and 8, so it's P680. That's how I remember it. Now, photosystem 2 comes first, and then it's followed by photosystem 1. And that is because photosystem 1 was discovered before photosystem 2, so it kind of threw off the whole order. Just know that 2 comes before 1. Okay, so here's the process of the light-dependent reactions, also called non-cyclic photophosphorylation, and you're going to understand why it's called that in a second. So here we have the thylakoid membrane, here we have the stroma, and here we have the thylakoid lumen. And here's our photosystems, photosystem 2 and photosystem 1. So water is first split by photosystem 2. Remember, photosystem 2 comes first in this process. This is going to release protons into the thylakoid lumen. The oxygen will be released into the atmosphere. This is what you're breathing right now. So these electrons from the water are going to get excited in photosystem 2 by sunlight, which are photons, right? And this makes the electrons go up into a higher energy level. They are then passed off to a primary electron acceptor. So this primary electron acceptor will then pass the electrons to an electron transport chain embedded in the thylakoid membrane. So what did the ETC do in cellular respiration? ETC, of course, referring to the electron transport chain. What was its purpose? Yeah, it was to pump protons. So this ETC will pump protons from the stroma into the thylakoid lumen against its gradient. So that's the purpose here. So both the lysis of water right here and the ETC create an electrochemical gradient, also called a proton motive force. Now, what protein synthesized ATP in cellular respiration? Yeah, it was ATP synthase. So plants use the same protein that we have in our bodies to make ATP utilizing this gradient. So protons are going to flow down this gradient and drive ATP synthase to help catalyze the reactions of ADP to make ATP, adenosine triphosphate. This is how plants make ATP for photosynthesis. And all this ATP made is just used exclusively for photosynthesis. It's not used for other cellular processes within the plant cell, just for photosynthesis. Okay, it doesn't just stop there. Now the electrons from our initial water molecule are going to be used again. So they're given to photosystem 1 next. So remember, photosystem 2 comes before 1. And photosystem 1, after it receives these electrons, is going to use sunlight, or photons, to excite the electrons back up to another higher energy level. When this happens, the electrons will be passed to another electron acceptor and travel down another ETC. The difference here is that the final protein is different. It isn't ATP synthase. So remember what I wanted you to memorize about the light reactions, about photosynthesis in general. What's the function of the light reactions? What are the products that are made? Remember, it's ATP and NADPH. And we have already made ATP. So the last protein of the second chain is called NADP plus reductase. So this protein gives electrons to NADP plus to make NADPH. So that's how we get ATP and NADPH. So now we have ATP and NADPH that can be used in the Calvin cycle. So see how this process isn't really a cycle. It's more of a one-way route through chemical reactions, hence why it's called non-cyclic photophosphorylation. Before we look at the Calvin cycle, let's learn about cyclic photophosphorylation. So this is less tested on, but it could come up. It's pretty rare, but it could come up. So in cyclic photophosphorylation, only photosystem 1 is used. The electrons energized in photosystem 1 are going to be recycled back into the first electron transport chain to make more ATP instead of being dropped off to make NADPH. Hence why it's called cyclic photophosphorylation. So the purpose of cyclic photophosphorylation is to pump more protons into the thylakoid lumen to make more ATP. That's essentially all you need to remember for this process. Now let's look at the Calvin cycle, or the light-independent reactions. These are called this because they do not directly use light energy or photons, but can occur during the day even when light is present, not just at night. So the Calvin cycle is dependent on the products of the light-dependent reactions. So without light, the Calvin cycle would stop because it needs ATP and NADPH. The Calvin cycle takes place in the stroma of the chloroplast, and it fixes CO2. Now CO2 is an inorganic molecule. Fixation refers to taking this inorganic CO2 and incorporating it into an organic molecule that can be used in biological systems. The organic molecule is glucose, C6H12O6, and in order to make this carbohydrate, 6 carbons here are needed. So plants need to fix 6 CO2 molecules. Thus, the Calvin cycle needs to repeat 6 times, hence why it's called a cycle. So let's walk through the steps of this cycle. So first things first, CO2 is going to combine with a 5-carbon molecule called ribulose 1,5-bisphosphate, or just RUBP for short. And that's going to make a 6-carbon molecule. So this 6-carbon molecule will break down into 2 3-carbon molecules called phosphoglycerate, or just PGA for short. So this process is done by an enzyme called rubisco. And fun fact, rubisco is the most abundant protein here on Earth. So next step is we're going to use ATP and NADPH that the plant made in the light reactions. So PGA is going to be phosphorylated from ATP, and then it's going to be reduced by NADPH to make G3P, or glyceraldehyde 3-phosphate. So this ADP and NADP plus will go back into the light reactions and become ATP and NADPH again. Now most of the G3P will be converted into ribulose 1,5-bisphosphate, or RUBP, with the help of ATP, but not all of it. The remaining G3P is going to be used to make sugar. So again, this process is going to take 6 rounds to complete. So it's going to continue 5 more times to fix all the carbon needed to make just 1 glucose molecule. It's quite the process to make a carbohydrate. So other carbs can be made too, like fructose or maltose. And then these monosaccharides can be combined to form disaccharides like sucrose, and then polysaccharides like starch and cellulose, and so on and so forth. But the overall chemical formula for the Calvin cycle is as follows. It's unlikely that you will have a question on these numbers here. Just appreciate the complete process. So again, it's 6 CO2 molecules plus 18 ATP plus 12 NADPH is going to yield 1 glucose molecule.